Mendel's experiment on inheritance. Gregor John Mendel is the father of genetics. He was an Austrian monk and he lived in monastery, and there was a beautiful garden outside, and there were garden pea plant, that is Pisum sativum, where he studied the inheritance in them, and he did some cross breeding and producing the hybrids, monohybrids, dihybrids, and he did various experiments and studied the inheritance, how the inheritance took place, and he did a great job, and he gave the basics of inheritance which help the people to breed animals and get the maximum profit, or to get the characters which they want in the progeny. So let's see what he did. Now he selected the garden pea, which had many different characters, different traits of characters he selected, and he bred to produce the new form. So it helped. Pisum sativum is the vatana. You remove the shell and take out the vatana out of it. So that how he studied the various characters. Now the characters here he studied is the flower color. Color were different. There was purple and there was white. Then seed color, yellow, green. There was seed shape, round and wrinkled. Pod color, that is the cover, that is green or yellow. Then the pod shape, it was inflated, that is puff type, and the other what was constricted, very tight. Then the flowers, position of the flower, that is terminal and axillary. Then the height of the plant, tall and short. So these are the 8 characters which he studied in the garden pea, and studied how the inheritance of this character takes place. Then he crossbred the different traits, the traits of the character, and produced new plants that form the basic of the inheritance, and he gave different laws on it. So let's see what he did while breeding. Now what he took, 2 plants. Now this plants, terminal flowers, axillary flowers, they self-bred, means like the pollens that fell on the carpels, and the zygote was formed, and that had the character, same character, generation after generation. Now why he took Pisum sativum? Because they had very small lifespan, so it was easy to study many generation, and they self-bred, therefore they were pure form. Like this terminal flowers, generation after generation they were having terminal flowers only, and axillary flowers, plant was producing axillary flowers only. So here alternative form, varieties which had alternative form of one characters. So here alternative forms in the characters was present in the garden pea, Pisum sativum. Then pure forms that bred. So this pure forms like the terminal flowers and axillary flowers, generation to generation, they were pure forms. They had the same traits of that character. Now they self-pollinated. Means in nature, the stamens, the pollen grains, they fell on the carpel, fertilization took place, and the zygote was formed. So self-pollinated, but they could be cross-pollinated. Like the pollens from this terminal flowers could be poured on the plant which had axillary flowers. So cross pollination could be done with some exercise, and the lifespan of this plant is very short, so you are able to study many generations. So what he did was, this is terminal flower. Terminal is the flower which is there at the end of the plant, and this flower was produced in the axil, not at the end. Before that, in the axil, the flower was produced. So these are the different traits of that character in the plant. What is the character? Production of flowers. What is the trait? The trait is, here it is terminal flowers, and this is axillary flower produced in the axil. So these two traits of the same character present in this parent. This is terminal, this is axillary. They are pure breeds from generation to generation. They have been this axillary, this terminal. Now what he did? He took the pollen grain from terminal flowers, pollen grain, and the ovule of the axillary flower. So when he had poured this pollen from terminal flowers on axillary flowers, what the first generation, that is filial generation, produced here is all axillary flowers. Why? Because this is dominant. This is recessive. This is recessive, this is dominant, and therefore the next generation is having both the characters. That is, this is dominant, this is recessive, both the genotype is same. All the first generation hybrid. Now this is hybrids are produced. First, this is first filial generation of hybrid, that it is AA. That is, it has got genotype, heterozygous genotype. That's two different traits are present in the genotype. But phenotype, all will have this axillary, because axillary is dominant. So the phenotype axillary. Outside they will all have this axillary flowers. Flowers present at the axil, because that is dominant character. But this recessive character is present. Now this filial first generation, this is the genotype. Now this gametes are separated. Gametes are separated. So this is capital A axillary, and this is terminal. Capital A axillary, and this is terminal. So now these gametes are separated. So what type of combination is possible? That is AA. Now this A will combine with AA is produced. So now this small a, that is the terminal flowers, is combined with the capital, this AA is produced. This A combined with the small a, big A, small a is produced. Now this small a combined with this small a, this becomes AA. So now the chances are this is capital A small a, capital A, small a, capital A, small a. This is 3, this has got two traits. Dominant trait is having axillary flowers. So all the 3 will have axillary flowers, and only one type, one out of this 4, only one will have terminal, because they have these two recessive traits coming together. So three will be auxiliary and only one will be terminal. So this ratio is 3:1. This is the monohybrid cross which has obtained 3:1, 3 auxiliary and 1 is terminal. So this is one experiment which he did to produce this monohybrid cross. Now he took for height, so pure tall TT, dwarf TT. Now when the gamete formation takes place, they will segregate TT, and this recessive character small t will separate into TT. So this is female gamete which is small t, that is for shortness. Big T that is for tallness, that is male gamete. So let's see what type of combination is possible. This small t combined with capital T. This small t combined with capital T. So here this small t, capital T, small t, capital T, small t. So these are pure breeds. That is, they have the recessive trait. Both have recessive trait. Here they have the dominant traits, that is capital tallness, capital T. So now the next generation all will be tall. Their phenotype is for tallness, but the genotype will have dominant as well as recessive genes. So this combination is possible. All the progeny will be tall. Now this filial first generation, now when they breed, then what combination forms in F2 generation? This is the second filial generation. So here combination of this, let's see, T, capital pure breed tallness, capital T, small t, capital T, small t, T, small t, capital T, small t, small t, small t, small t, small t, both are recessive. Therefore in progeny 3 will be tall and only 1 will be short. So again we get this character in F2 filial 2 generation. You get 3:1. 3:1 phenotype. Now genotype, it is 1 is tall, 2 are heterozygous and 1 is short. So genotype ratio 1 to 1. 1 to 1. Here also, here the genotype, here only 1. These two are same. So 1 to 1. That is the genotype. Whereas phenotype, what you see the character in the offspring, it is 3 auxiliary and 1 terminal. So it is 3:1. Same way for this height, 3 phenotype, what you see in the next generation, 3:1. What is actually there in the genes, genotype, it is 1, 1, 2, 1, 2 and 1. So 1, 2 and 1. Whereas 3 will be tall and 1 will be short, because they have got this recessive genes. So by studying, Mendel gave this law of dominance. That is, there are 2 traits for one character, and one is dominant and the other is recessive. Whichever is dominant comes in the progeny, and whichever is recessive does not show in the phenotype. So supposing it is tall and short, tall and short, then the progeny is going to be tall. Small t, shortness, is not shown in the progeny. So what is dominant? Tallness. Recessive is shortness. So according to the law of the dominance, there are two traits which are present, and whichever in the for a character, and whichever is dominant comes in the progeny or is shown in the next generation. And the recessive trait is shown in the progeny, in the phenotype, when both the traits are recessive. Supposing both are small t, then the progeny will be short, because here the both are recessive. If one is dominant and the other is recessive, the progeny, the phenotype will be tall. And if both are dominant, then the progeny is tall. So this is law of dominance. Now, law of segregation. Whenever the gamete, now this is the female gamete, small t gamete, there is segregation when the formation of gamete takes place. Now our cells are diploid. Whenever the sex cells are formed, they become haploid. That is, my XX will be separated. If there is a male XY, then XY are separated. So there is complete set of 46 becomes 23. So complete segregation takes place in the gamete. That is applicable for the characters. Now this female gamete TT has separated into TT. This is the male which has separated into gametes. So this TT will be separated in the gametes. So when the formation of gametes takes place, then the characters are separated. So that is the law of segregation. The traits are separated. So whatever the combination takes place in the zygote is random. So whatever it is, just by chance what the combination takes place. So here it is pure form of TT, but here it is T, capital T and small t. So what will happen? It is random chance whether this combines with TT or this TT or TT. It's random because segregation has taken place, and it is random by chance what combination takes place in the zygote, because gametes are haploid, zygote is diploid. So now when it becomes diploid, whatever the characters are dominant will come in the zygote, comes in the offspring.